Don't clap yet. You haven't heard what I have to say. So I'm Dr. Declan O'Rourke. I'm a pediatric neurologist in Temple Street. I have a particular interest in neuromuscular disorders. Consider myself the lead in pediatric neuromuscular disorders in Ireland in terms of care coordination. I was asked to talk to you today about this particular study, the DYNE-DELIVER study. It's a phase 1/2 study that we are conducting in Temple Street. It's part of a multinational pharmaceutical-led study from DYNE Therapeutics. This is our first early phase clinical trial in child neuromuscular disorders in Ireland. It's actually our first early phase clinical trial in pediatrics that we have also conducted. So I was delighted to be able to talk to you about this. These are my disclosures, and I suppose the only thing that I want to highlight in terms of my disclosures is that we have been involved for some time in quite a number of clinical trials in Temple Street, all related to neuromuscular disorders. All phase 3 studies. So we were I suppose we had a track record and been able to deliver phase 3 studies, and hence probably why we managed to be successful in being selected as a site for this early phase study, which is important work. I suppose the complexities around running an early phase study is that it's essentially a first in human trial. It's the first time drug would have been delivered to patients. The study is designed in terms of assessing safety rather than efficacy, but there's some efficacy outcome measures in relation to this. And again, because this was our first time performing an early phase study, we collaborated, and we are collaborating with our colleagues over in this clinical research facility in St. James's to assist us with this, where they have a record of performing other early phase clinical trials, so we're utilizing their experience. And we have some other collaborations on the go in relation to neuromuscular care for spinal muscular atrophy patients that we're delivering in the CRF in St. James's. So their help and assistance in this seemed to be a natural transition. This was an extremely challenging trial to open. From the time we were initially approached from DYNE Therapeutics to when the study was first opened at our site and we got to recruit our first patient for the study, there was 18 months had passed. A lot of that was down to repeated substantial amendments, all of which needed to go to central ethics, be reviewed. Another substantial amendment would come in. As a site, when you're being open as a clinical trial, you're very much managed by the sponsor in relation to this and the CRO involved, but there's a lot of moving parts in getting set up and getting open for a clinical trial. This one was especially challenging. So this is a study looking at a compound that to affect the dystrophin production in boys with Duchenne muscular dystrophy. So Duchenne muscular dystrophy, life-limiting disorder. Typically, patients lose ambulation between the ages of 9 and 12 years of age. And with some intervention, specifically steroids, cardiac medicines, and ventilation, life expectancy improves, but there's real unmet need. In 2005, very limited therapeutic targets. There's really been an explosion over the last 10 to 15 years. And so this study is looking at utilizing a compound involved in exon skipping. So what's exon skipping? Well, just to give you an insight into that, the dystrophin gene is the largest gene in the genome. Most boys who have Duchenne muscular dystrophy have a deletion somewhere within that gene that leads to an out-of-frame mutation. So this patient, for example, is deletion exon 48 to 50. So you can see that exon 47 doesn't fit into exon 51. Compare that to this boy who has Becker muscular dystrophy, whose deletion in 48 to 51, where 47 now fits into 52. So going back to our boy with the 48 to 50 deletion, if we were somehow able to skip over exon 51 by using an antisense oligonucleotide technology treatment, which is what we're doing in this study, we would be able to restore the reading frame where 46 can now fit into 52 and allow some dystrophin to be produced. Similarly, in this case where if we could skip 45, 43 would fit into 46, and in this case 44 or 43 would again fit into 46. So this study looks at utilizing a PMO, a phosphorodeiamidate, morpholino oligomer, which is an antisense oligonucleotide treatment to exclude exon 51 to allow in pre-messenger RNA reading to allow protein translation to occur. In our patient population, that would account for about 12 of our patients. About 13% of our population have a pathogenic variant in the dystrophin gene amenable to exon 51 treatment. What's novel about this, and there are other exon 51 treatments out there, some of which are FDA approved and commercially available, eteplirsen being the main one for exon 51. What's novel about this is that this is using a specific antibody attached to the antisense oligonucleotide that will specifically bind to the TFR1, the transferrin receptor 1 on muscle to allow increased specificity, increased uptake within the muscle that brings it to the cell nucleus to allow the antisense to exclude exon 51 treatment. There's preclinical modeling that shows in MDX mouse and other animal models dystrophin expression with its use. So studies looking to enroll 88 boys globally, about 30 sites. The primary objective is regarding safety and tolerability, and then dystrophin protein expression. So that involves doing muscle biopsies on patients at the start, and then at a specified time point during the study. So two muscle biopsies. There are secondary endpoints looking at pharmacodynamic biomarkers, muscle functional outcomes, PK, immunogenicity. The muscle functional outcomes include outcomes for both ambulatory and non-ambulatory patients, as well as some quality of life assessments. One of the reasons why we were keen to get involved in this study is that it allowed us to recruit patients who were with Duchenne muscular dystrophy who were non-ambulatory. There's a real unmet need for exploring therapeutics and allowing boys with Duchenne muscular dystrophy who are non-ambulatory to be able to participate in clinical trials like this, and this study has allowed us to do that. The dose optimization involved a multiple dose escalation, starting off from a small, 2 mg per kilo, escalating with each separate cohort of multiple dose escalation up to an aim of 88 mg per kilo. The age for inclusion is boys between 4 to 16 years of age who have a Brooks upper limb score of 1 or 2. So that means that boys have to be able to lift their hands over their head. When we look at our overall population who might fit into that, we've actually only had 1 or 2 patients who have been eligible to be included in the study. There's some additional inclusion criteria in terms of respiratory and cardiac status, some exclusion criteria specifically related to cardiac status and the delivery of other dystrophin restoration therapies, whether in trials or commercially available. So there is some data readout already on the study at the 10 mg per kilo and at the 20 mg per kilo. Our site was managed to be opened at the 40 mg per kilo, so we haven't yet got data readout on where our site are at, or the patients that are participating in the study from our site. But there's 10 mg per kilo and 20 mg per kilo data readout. These are some of the demographics here in relation to that. I suppose one of the things to note that acute kidney injury and thrombocytopenia was seen in a couple of patients, which required a brief pause in the study to assess. Ultimately, things have been allowed to progress. Fever, headache, and vomiting were other commonly related side effects. So what we're seeing is impressive exon skipping, robust exon skipping, and robust dystrophin production in biopsy compared to baseline. I suppose the company have looked to comparatively, look at this data compared to other similar products. But again, it's early days in terms of data outcome for this study. But there is a signal, certainly in terms of robust dystrophin production, which is the primary endpoint for this study. There's also a secondary endpoint in using the SV95, which is the stride velocity, assessing them at the 95th centile. This is a novel outcome measure tool in muscular dystrophy trials and in neuromuscular trials, where it can assess the stride velocity using a device, a little bit like an Apple Watch that's strapped to the ankle, and again some signals towards improvement in stride velocity from time to rise from the floor and in 10-meter walk, test, run, but it's all over a relatively short time period. So summary, the data regarding safety appears favorable. There does appear to be robust expression of dystrophin. There does appear to be robust expression of exon skipping. We're getting some signal towards functional outcome measures. And then the next rollout in terms of data is 2020, or the end of 2025. It's a 24-week study that then runs into open label extension, that then runs into long-term extension. The aim for long-term extension is 4 years. That's us in the middle as a site, along with the rest of the sites involved globally. I suppose, as I said in the beginning, we were very happy to be involved in this translational work, and it was the first early phase clinical trial in pediatrics that we've been involved in Children's Health Ireland, and so we've been pretty proud to be part of that. Our co-location on the new Children's Hospital, when it opens, gives us this advantage geographically with how we are going to be closely located to our colleagues in the clinical research facility on the St. James's campus, that will allow us to continue to work together to be able to perform these type of early phase clinical trials, and we're hoping to now that we have one, to acquire more. So I want to acknowledge our research and innovation teams that really worked very hard to get this study open. The CRF clinical, or the research facility team, my own neuromuscular clinical and research teams that work hard on this trial and other trials that we're involved in. The executives that were, and I mention those because we are a children's hospital collaborating with a very adult hospital, and so we've needed to have executive buy-in from both hospitals to make this happen, and of course our industry collaborators. Excuse me. Thank you. I'm happy to take questions, but if not, move on.
Are there any questions to be had just now?
No, again, the wearability of the device seems to be quite comfortable. Again, I describe it similar to wearing an Apple Watch. We've had no patients complain of any friction-related issues. It does seem to be. Oh yeah, I mean, like patient compliance in that. I suppose we have our experience is N of 1 at the moment, whose tolerance has been quite good to this. We've not had feedback that tolerability or compliance has been an issue. So you know, presumably it's an easy enough device to wear. The weight is quite low. You know, I think patients are getting used to it. Yeah.
Other questions?
No, it's not. Hi, really impressive work. I was just wondering, what is the rationale underneath the fact that one of the exclusion criteria is the age? So you just put on your clinical trial under 13 years old, is that so?
So the age range for this study was boys older than 4 and less than 16 years of age. There was an ambulatory and a non-ambulatory arm. One of the nuances regarding the non-ambulatory arms is that boys had to have lost ambulation within the last 2 years. So if somebody had lost ambulation, was say 15, but had lost ambulation at 12, they were excluded from the study. The attraction from our perspective and from patients' perspective is that this is one of the few studies that has offered inclusion of patients who are non-ambulatory to be able to participate in a trial like this. Again, the study is largely focused on safety and demonstrating dystrophin expression as proof of concept or additional proof of concept, rather than being focused on outcome measures that will hopefully come later in a phase 3.